Several years ago, researchers in northern Sudan discovered the skeleton of a young boy from ancient Egypt who suffered from cancer. While there had been some hints in the archaeological records that cancer isn't just a modern disease, finding such an old skeleton riddled with holes caused by some type of cancer was shocking, to say the least. Nonetheless, this incredible find is helping the research community today to better understand the mechanism that causes this terrible disease. Unfortunately, 3,000 years after the young boy's death, we are still searching for ways to eradicate this threat. According to the American Cancer Society, 1 out of 3 people in the United States will develop cancer during his or her lifetime. Current cancer treatments, such as chemotherapy, for example, are harmful to the whole body, causing side effects like hair loss, fatigue, depression, and infertility, among others. Other treatments, such as surgery and radiation therapy, are too localized to be effective against cancer that has spread to other parts of the body. Thus, there is a need for designing new drug delivery systems for safer cancer treatments. Since cancer cells send out angiogenic factors to encourage new blood vessels to grow and to send more oxygen and nutrients into the tumor cells, the tumor grows rapidly. The blood vessels around the tumor then begin to leak, keeping the much-needed nourishment from reaching the healthy cells. Recent advancements in nanoscience and nanotechnology have given rise to promising biocompatible lipid-based drug delivery systems that utilize the dual water-loving and water-repelling characteristics of the leaky blood vessels around the tumor and use them for cancer therapy. Among others, the drug delivery systems include liposomes and nanodiscs. Liposomes are spheres composed of a bilayer of lipids with a water-filled core that can encapsulate or enclose both water-loving and water-repelling cancer drugs. Nanodiscs are very small disc-like particles composed of a bilayer of lipids that are able to enclose water-repelling cancer drugs. Additionally, the systems can be modified by adding ligands, which are molecules that can seek and locate cancer cells, so the systems can be better at releasing the drugs. This and other innovative research is being done in many laboratories around the world. Such is the case in Dr. Mu-Ping Nieh's lab at the University of Connecticut. Dr. Nieh and 2 of his graduate students, Chung-Hao Liu and Donyeil Hoy, are researching and testing a variety of delivery systems using nanodiscs, also known as bicels. Although liposomes can be created with one type of lipid, preparing bicels requires more than one type. Bicels require 3 types of lipids, one with a head and 2 long chain tails, DPPC, one with a head and 2 short chain tails, the HPC, and one with an electrically charged head and 2 long chain tails, DPPG. Like in cooking, the ratio between the lipids is important and must be carefully calculated. To enclose or encapsulate a cancer drug, lipids are added along with the drug into the mixture. The mixture is then heated and cooled several times. The result is a group of bicels with the drug inside their bilayer of lipids. Aside from controlling the ratio between lipids, it is also important to control the size of the liposomes. A tool called an extruder is used to filter out particles that are too large to be useful. Because the bicels are so small that they can't be seen with the naked eye or with a conventional microscope, other methods must be used to determine the characteristics of the nanodiscs. In Dr. Nieh's lab, many different techniques are used. For example, the size of the bicels is estimated by using dynamic light scattering, or DLS, a technique where the larger bicels in a fluid move slower and the smaller bicels move faster due to the random movement of the particles as a result of their collisions with each other. UV-vis spectroscopy is a technique used to identify successful drug encapsulation inside the bilayer structures by measuring the amount of light that is absorbed by the bicells at certain wavelengths. It is also used to measure the percentage of drug that is encapsulated inside the bicells. Fluorescence spectroscopy is also used to identify successful drug encapsulation. However, rather than measuring the amount of light absorbed, it measures the amount of light emitted. If the sample does not fluoresce, then there is no encapsulation. This is because pristine bicells do not absorb light in the ultraviolet and visible spectra. It has also been found that the emission intensity is greater after the drug has been encapsulated as compared to the emission intensity of the drug alone. However, the reason behind this is still unknown. In the lab, in order to confirm the morphology or structure of the samples, students can take images of the bicells using transmission electron microscopy or TEM. It is impossible to measure the size of the bicells by using copper grids of a predetermined size. The picture also shows whether bicells were created or if much larger spherical molecules known as vesicles were created instead. Another way to confirm that either bicells or encapsulated bicells have been created is to use small angle X-ray scattering, or SAX, to determine the size of morphologies of the samples. Finally, once all the testing is done, lung cancer cells are mixed in solution with the encapsulated bicells in a drug study to see how the cancer cells react to the new drug delivery system. If the new delivery system proves successful, it will be tested on cancer tumors on rats. If that succeeds, the system could then be proposed to be used in clinical trials using human cancer patients. It should be obvious by now that the next generation of drug delivery systems needs more brilliant engineers. You could be among the future men and women who work diligently to eradicate cancer and work to help save thousands of lives. Maybe there's even a Nobel Prize in your future. We would like to give a special thanks to Dr. Mu-Ping Nieh and his doctoral and postdoctoral students, Chung-Hao Liu, Donyeil Hoy, Dr. Kuo-Chih Shih, and Farnoosh Saeedinejad for the opportunity to spend a summer learning about this very important project.